Where is everybody? Sickness? I know a few people are sick, but all right. So exam on Friday. So my plan is to get here 1:30ish. And I think last time I asked, some of you said you could get here early. So, and then I'll stay late as well. I think I can do that as well. So, and then Duncan will be the other person here. So it'll be the same basic idea that you'll have two versions of the exam, multiple choice and essay. And then before you leave the room, just make sure to show either Duncan or myself that, it's all submitted. Questions on exam stuff? Obviously, it'll be pretty heavy physiology, but the cell structure and function too. There'll be definitely questions on that. All right. Well, I will be checking email frequently. So if you're studying, you have a quick question, I can answer that in a timely fashion. Right now there's two, but I have not finished making the exam yet, but two for sure. I don't think I'll back that down to one. So for sure two. And one is coming from cell structure and function. The other one's coming from physiology. And I'm debating about a couple of smaller ones I might add in there, but I haven't made up my mind yet. Okay. So this topic that we're starting right now will not be on the exam. It will be on exam 3, I guess. And so this is the idea of photosynthesis. So I know some of you have had plant classes where they have talked about photosynthesis. So if you've had anything like that, you're going to be in good shape for that portion of it. But in the microbial realm, some microbes do photosynthesis pretty differently compared to plants. And so the way I'd like to teach this is to get you oriented with at least, I think most of you know some vague idea of photosynthesis, like how plants do it. So I'm going to talk about that first. And then we'll get into some variations on how some microbes do this differently. So when I say plants, I'm also considering, so plants, their version of photosynthesis, cyanobacteria also do pretty much the same thing. And so cyanobacteria are very similar to how we teach about photosynthesis in our plant classes. So we often show this idea. This is kind of a summary statement figure of photosynthesis. So when we talk about photosynthesis, we break it down into the light reactions of photosynthesis, and then the so-called biochemical reactions. So you can see those written down on the bottom there. So with the light reactions, that's mostly what we're going to focus on for this version of the class. We're going to look at how some microbes do it differently. But basically, this figure is combining all of photosynthesis. It's looking at light reactions plus the biochemical. When they say biochemical, they mean what does the plant do with the carbon dioxide that it's bringing in and that's making sugars off of it. And so the biochemical reactions are what happens to carbon dioxide. So what you're seeing here is the combination of both the light. So on the left, you can see they have, well, the whole picture is a chloroplast, and they've cracked it open so you can see what's going on inside, and they're showing how light energy is being absorbed by the internal membranes inside the chloroplast. And as that is occurring, they're trying to show that because of that process, you end up with some really important molecules, ATP and NADPH. And then they're trying to show how those two molecules feed into the biochemical reactions, which are often referred to as the Calvin cycle. And the Calvin cycle is the process where the plant will take in carbon dioxide through its stomata on its leaves, bring it internally into the cells, to the chloroplast, and then you can see this is a cycle, so it goes round and round and round all day long. And what they're trying to show is there's a key molecule in the Calvin cycle that gets sent off into another pathway in order to make sugars. And if you look at how plants make their sugars, they don't care about glucose in this regard. They're after sucrose. So they're trying to make sucrose through all of this. Okay, so this is a very big picture idea. If you look at how plants do it, how cyanobacteria do it, how some other microbes do it, there's some common themes. You have some kind of so-called antenna system, which I'll explain. You have these so-called reaction centers. You have some version of an electron transport chain process. And you also have energy carriers. So it doesn't really matter who's doing the photosynthesis. This is all going to happen. So I like that they named it antenna system, and at least if you're familiar enough with the old-fashioned antenna, you know, the rabbit ears that you put on your TV to capture better TV signal. That's what I use in my house still. And so it's analogous to that idea, where there's going to be these pigments that form these complexes, and they're busy capturing sunlight energy and absorbing it. And so they're absorbing these different wavelengths of sunlight energy, and then they deliver that energy to where it's needed, which is the next bullet, the reaction center. Right? So the antenna complex is like, okay, I'm going to absorb all this different wavelengths of light, and then I'm going to funnel that to the reaction center. And the reaction center is named the reaction center because there's going to be the kickoff to a biochemical reaction there. That's really important to essentially kick off an electron transport chain process. So in the reaction center, all that light energy gets down, well, we often say it gets funneled down into this reaction center. That provides the energy to do a couple of things in that reaction center, with the end result being you get the kickoff of an electron, zinging down an electron transport chain from donor to acceptor. And then those energy carriers, you've already met some energy carriers when we talked about the TCA cycle. And we said, you know, NADPH gets created, FADH2, NADH is another one. Those are all energy carriers. So we need to think about those two. They'll be involved in these aspects. Okay, so these are kind of like, okay, they all have that in common. So the antenna system, they're actually trying to work it out, like what does it really look like down at the molecular level. And so what they're trying to show here is how these antenna systems, they label LH2. LH just stands for light harvesting. They're harvesting, absorbing light energy coming from the sun. And what they're trying to show is these antenna complexes surround, right in the middle, is the reaction center. Right? So it makes sense that the antenna systems are surrounding that reaction center because the antenna systems are absorbing light energy and they're passing it to the reaction center so that the reaction center has a constant supply of that sunlight energy to do what it needs to do. All right. So if you've learned anything about how plants do photosynthesis, you might see something like this. So this is a common way to depict it. And again, this is combining light reactions and the making of sugar using the Calvin cycle as part of that process. So if you look at plants and their leaves typically, and lots of chloroplasts inside their leaves. Remember, chloroplasts were once free-living microbes that have been put into the eukaryotic cell type. If you crack open the chloroplast, you'll see an internal membrane that they call the thylakoid. I would like you to know that term, please. So the thylakoid membrane is this internal membrane floating around inside the chloroplast, and that's where the light reactions are taking place. You have all of these complexes that they've drawn out here. That membrane that you're looking at, that they're embedded in, that's the thylakoid membrane. Okay, that's the thylakoid membrane. And so you have lots of players, and again, they're showing this arrow pointing, going from all these different players in the game. So you should start going, oh, that kind of looks like an electron transport chain process. And you would be right. So when plants are doing photosynthesis, excuse me, we talk about these two photosystems. Plants have two photosystems. We call them photosystem 1 and photosystem 2, Roman numeral 1 and 2. So you need to know that. And then a couple of things happening here to pay attention to. With photosystem 2 on the far left, you see that water is going through a reaction, and it's converting water over to oxygen and protons. So you see those are the products, water, and then it's swooping over and it's going, you're in this photosystem 2 reaction, you get the products of oxygen and protons. But there's another thing super important that is coming off of water, part of water, and that is electrons. Right? So electrons, E minus, ugh, doesn't want to do it. So E minus, that's supposed to be an E minus. Electrons are getting stripped off of water. This is incredibly difficult to do. Water does not want to give up electrons. So this is really, really challenging. You need a powerful light energy source. You need a very special complex embedded as part of photosystem 2 to make that happen. And so if you're going to strip electrons off water, you are going to end up with oxygens and protons. And then that electron is getting sent through this electron transport chain process. So what we say when plants are doing photosynthesis, we say that water is the ultimate electron donor. You need to know that. Water is the ultimate electron donor. Because we're going to find that some microbes don't use water as an electron donor. Okay? So if water is being used as an electron source, remember, just you have to know that if you strip electrons off water enough times, you're going to end up with oxygen and a bunch of protons. So that's how we're able to exist, because cyanobacterian plants are busy doing this version of photosynthesis that gives us oxygen so the aerobic organisms like ourselves can survive. Okay. The electron transport chain process, you see it's going through a series of players embedded in the membrane. And eventually you do also see photosystem 1 will get those electrons. But you see photosystem 1 is also getting hit with light energy. So both photosystems, you have antenna complexes around both of those. You have those antennas absorbing light energy, giving it to the photosystems. The photosystems also have pigments inside them, so they're absorbing energy. So you have these two photosystems, and they're linked through this chain, this electron transport chain. Right? So there's two photosystems. So how many photosystems are present is important, because we're going to see that varies. And then you see here that NADP plus is sitting at the end of this process, and as it accepts electrons, it becomes reduced to NADPH. So we say that NADP plus is the terminal electron acceptor, or the ultimate electron acceptor. And that's important to know as well. We're going to see that can vary. All right, so NADPH now that's fully reduced, so that's a reducing agent, reducing molecule. That's good. So an electron transport chain process is cooking along as soon as the sun strikes the plant, and this is happening all day long. And so if this is happening all day long, water keeps getting stripped of electrons, you get a bunch of protons starting to build up. And you have more protons on the lumen, the bottom. If you crack open the thylakoid membrane and look inside of the membrane, that's called the lumen, and that's what's being shown down low. They say thylakoid interior, but the real word for that is lumen, L-U-M-E-N, just like our intestines. When you look inside of our intestines, it's called the lumen. The stroma is the watery portion inside the chloroplast. So you have the lumen separated by the thylakoid membrane to the stroma. You have protons developing in the lumen area, more protons there, less protons in the stroma. So now you can see that there's a proton gradient that is getting established across a membrane. Anytime you see a proton gradient across a membrane, you should say, hey, that could set up a situation to make ATP. And that's what they're showing here. They finished the thought where they put the ATP synthase there. And you can see that the protons are diffusing through there. And as that happens, it creates the situation where ADP can get phosphorylated. And you get that wonderful ATP. So some similarities to what we talked about with aerobic metabolism, anaerobic metabolism, where you have an electron transport chain linked to proton pumping and development of a proton gradient, which can then lead to ADP getting phosphorylated to make ATP. So this version of how the plant is making ATP is called photophosphorylation. All right, so this is the third mechanism. So when you look at all the ways organisms can make ATP, we have that substrate level phosphorylation we already talked about. We have oxidative phosphorylation. Now we have photophosphorylation. And the phosphorylation is talking about you got to get ADP phosphorylated in order to make ATP. Questions on this concept? Have most of you had some basic photosynthesis training? Some point? Maybe a little? As a dim memory? All right. But, we've already covered this idea of an electron transport chain. We've already been through this theme and the proton gradient because of that. So it's, you're starting to get this theme going of how these different organisms are playing the game in order to get their reducing molecules and their ATP. So this next slide is pretty much what we just talked about. So the fancy word for what we were just describing is oxygenic photophosphorylation or photolysis. And so oxygenic creation of oxygen. And the only reason we say it's creation of oxygen is because water is being used as a source of electrons, the ultimate electron source. And so once you strip that water molecule of electrons, you're going to eventually get oxygen. And so cyanobacteria were the first to do this before the plants. All right. First cyanobacteria were on the earth, and they did a ton of this before plants even could show up. And so cyanobacteria are, very important in the oceans as well for doing photosynthesis there. And they have their versions of photosystem 1 and photosystem 2, like what we see in plants. And they're using water to strip electrons off of, so that's their source of electrons. They're creating a proton gradient. And that proton gradient then can be used to make ATP. So they often refer to how cyanobacteria do it. They say there's 3 ATP and 2 NADPH per 2 water molecules that have been stripped of electrons. Photolyze just means you use light energy to rip an electron off of water, which is quite hard to do. And then why should we care about that? Well, it's because the ATP and the NADPH get used for part 2 of photosynthesis, biochemical reactions, in order to make all those sugars. Remember, chloroplasts that we talk about in plants, they were once free-living microbes related to the cyanobacteria, ancient microbes that came along and evolved this mechanism. So this picture is another version of what we looked at, kind of zooming in on how the cyanobacteria do it. But if you look at the lower image first, it's a little bit easier, I think, to see what's going on. You see that there's 2, oops. See that there's 2 photosystems. Photosystem 2 is on the left, far left there. Photosystem 1. And the light energy, under that squiggly line, that's the light energy. And there you've been showing the antenna system surrounding. And so the antennas are absorbing that light energy from the sun. They deliver it to the reaction center. This so-called chlorophyll A, here is, that's a special chlorophyll that's in the reaction center. And so what they're trying to show here is that you get the, well, let me go back to this. I'm going to shrink it down. You get the water being stripped of electrons. And then you can see I guess here they're using a blue line to show the flow of electrons from donor to acceptor, all the way down until you see the NADP plus down here accepting those electrons and becoming reduced to NADPH. So they're also showing the idea of the proton gradient. They're showing how the protons are getting pumped across the membrane. Water, of course, is donating protons to this. And so you get this proton gradient across the membrane, which allows for the ATP synthase to do its job and get ADP phosphorylated. This top figure is putting it in, they call it the Z scheme, but you don't need to know that. What this is doing is putting it on an energy scale, a reduction potential scale. So it's kind of like the redox tower that we were looking at in that regard. So the E in volts, negative. So there's 0 mark halfway, and then above it is negative reduction potential, and below it is positive reduction. So what they're trying to show here is that water is way down. Water's way down in the positive end because it doesn't want to give up electrons. Right? You have to force it with light energy. And when you do that, they're trying to show once you zap water with light energy and you strip electrons out, they show the electron go way up reduction scale here into the negative value. So what that's saying now, this electron is nicely excited and is capable of being given off to these other players in the game and go through this electron transfer. And then you're also seeing voltage 1 is kind of around 0, but it gets hit with light energy. And again, that excites an electron in the reaction. And again, going up into the negative reduction potential. So that means it's now that it's nicely excited like that, it's very easy to be given off to these other players. And then you finally see that your NADP plus will get it. And so that's just trying to put it into context, basically to show how light energy is really good at exciting these molecules and getting them willing to donate off electrons. Ferredoxin. So plants use ferredoxin, cyanobacteria as well. I would like you to know that. I know. That's one of the players in terms of getting an electron and its job is to pass the electron up to FNR, which immediately working with NADP plus. So FNR and NADP plus. FNR is an enzyme that will drive the NADP plus getting some electrons and protons. Okay. The next slide is just saying exactly what we've just been covered, so I'm going to skip that. It's all in words. Okay, so we're zooming into chlorophyll, whether we're talking about a plant or the cyanobacteria. We're using chlorophyll, and this is a chemical structure of it. They call this part in yellow. They call that the porphyrin head region of the chlorophyll pigment, and that has a magnesium ion chelated in the middle of the porphyrin head, and then they call this the phytol tail of chlorophyll. And so it's the tail region that actually sticks into the thylakoid membrane, spans the thylakoid membrane. And so basically these pigments are helping to absorb light energy. And then there's different kinds of chlorophyll. You have, they're trying to allude to that up here where you put T, they're saying C and X on the porphyrin head region here. That X is CH3, a methyl group. If it's like that, we say it's chlorophyll A. If you replace that methyl group with an aldehyde CHO, then it's called chlorophyll B, right? So there's different chlorophyll, and they vary slightly like that by their structure. So there's chlorophyll A, B, C, D, right? And so depending on the microbe or the plant or whatever, they're going to have these different chlorophylls. And that makes them absorb light a little differently from each other. So remember, light energy is different wavelengths of light. And then we often refer to those different wavelengths as different colors. Red, orange, yellow, green, blue, indigo, violet. Those are all different wavelengths of light. And these different chlorophyll pigments have favorite wavelengths that they want to absorb. And other wavelengths they will not even touch. Nothing happens if you hit them with that wavelength. And so the fancy way to say that is that last bullet point. These slight differences alter their absorption spectrum. That just means if you hit them with light energy, they preferentially absorb certain wavelengths of light over others. And a lot of others they don't even bother with. They're designed, evolved to deal with a certain wavelength of light. So chlorophyll A, we see those in plants. So when we talk about in our plant classes, we really do zoom into chlorophyll A and chlorophyll B, essentially. There are other chlorophylls that they've called bacteriochlorophyls. And that's only in the microbe. So we're looking at these so-called purple bacteria. That's a group of bacteria. They're actually called that. Purple bacteria, green sulfur bacteria, green non-sulfur bacteria, and then heliobacteria. So we'll talk about them. And they have bacteriochlorophyll. So their structure of chlorophylls is different enough that they gave them, okay, yep, they're bacteriochlorophyll. And then all photosynthetic organisms seem to have some flavor of accessory pigments that are part of the antenna complexes. And they're accessory. So their job is to capture different wavelengths of light and then get that energy to the reaction center. And so They can also serve a role to protect the organism against too much light coming in too fast. So too much of anything can be bad for you. Same with photosynthetic organisms. If they get hit with too much light too fast, it can start damaging their proteins, their membranes, their DNA. And so some of these pigments can absorb that too intense light and deal with it in a safer way to keep the organism from falling apart, essentially. Pretty much any photosynthetic organism, I don't think there is even an exception, but all photosynthetic organisms have carotenoids. That's a special type of pigment. Carotenoids, you're familiar with carotenoids if you eat carrots or other orangey, reddish orangey type fruits and vegetables. Those are chock full of carotenoids. And then phycobilins are another kind of special group of pigments that red algae can have, and then cyanobacteria can also. So what we're trying to say here is that it depends on the organ, what kind of pigment they're going to use. What did they evolve to use in order to capture sunlight energy? And it's highly dependent where they live, especially when you look at the microbes. All right, so microbes will have that live. So, for example, red algae, and then some other cyanobacteria that might live at little deeper depths of the ocean. Phycobilins are really good at capturing light a little deeper down in the ocean because the wavelengths of light that come through the water, those phycobilins are optimized to capture that light energy. And so it kind of depends where they live, which flavor of pigment they're going to pick. And again, they are evolving this based on where they live. They're using the process of natural selection. So here you can see the absorption spectra. So what they're showing here is that if you take chlorophyll A pigments, put them in a spectrophotometer, and run a scan, say I want you to scan this solution from 340 to 800 nanometers, and let's see what happens. And then you see these peaks. What that's saying is chlorophyll A is maximally absorbing light energy around what, over 400 nanometers, like 450, and then another peak right around 700. So that wherever you see the peaks, that's where it's like optimized. Like that's my favorite wavelength of light. I'm really good at absorbing that, and I can use that light energy for photosynthesis. Like the cyanide you see is slightly different, from 650 or so nanometers. And then the graph on the right, again, you're seeing these absorption peaks, but back to your chlorophyll and carotenoids, but you're seeing that in general, these different pigments absorb different wavelengths, let's put it that way, different wavelengths of light. And again, it just depends where they live. These different wavelengths can be, what they're surrounded by, what they can easily get access to. And so they evolve these pigments to take advantage. And so you can have a bunch of photosynthetic organisms all hanging out together, like in the water column, and they're fine with it. They're not competing with each other necessarily because they have different pigments. And so they're not all trying to get to the same wavelength of light. So they can happily coexist. And a lot of ocean-dwelling photosynthetic organisms do that. They'll live in consortiums, and they're not, competing for a certain quality of light. They can each have their own little share and be just fine. Okay, so lots of different options there. So here we're just looking at this idea of there are some microbes that will take in CO2 as part of photosynthesis to make their sugars. That's how plants do it. So plants are photoautotrophs, cyanobacteria are photoautotrophs. But there are some microbes that never evolved the ability to take CO2 and make sugars from it. So they have to get some carbon from an organic source, something that's dead and dying, decomposing perhaps nearby, and then they can take that carbon source and use it to get their carbon needs. Okay, so microbes can be a little murky on whether or not they're going to use CO2. It depends. So that's a big exception. If you've taken a plant class, we only talk about CO2. How CO2 is being used. Microbes, maybe, maybe not. Okay, here's another idea of what are the options in the microbial world in terms of photosynthesis. You're seeing two big ideas. Oxygenic is what we've been focusing on. That's the idea of water being used as the ultimate electron donor, a reducing power. That's what they mean. Okay. Water is being used as the ultimate electron source. And then they're trying to show a little drawing here of how electrons are getting fed into, photosynthesis, and you end up making ATP, and then they're showing CO2 is getting used to make sugars. Now compare that to the left side of this figure, anoxygenic. An in front of a word in science means without oxygen creation, is what's saying there, anoxygenic. So you don't see water as the reducing power. You see something else. In this case, hydrogen sulfide gas, which is a common option. There's a lot of sulfur in the environment, so hydrogen sulfide is pretty common. But again, it's the same basic idea in the sense that they're using hydrogen sulfide as an electron source, their ultimate electron source. It's just that if you strip electrons off of hydrogen sulfide, you're not going to get oxygen. So it's anoxygenic. But they're still doing their version of photosynthesis. They're still getting an ultimate electron donor, hydrogen sulfide, stripping electrons off of it, sending it through an electron transfer chain. In the process, there'll be a proton gradient. They're not showing that here, which allows for ATP. And then they're also showing the idea of CO2, in this case, being used to make carbohydrate. So this is another big picture idea of, in the microbe world, yes, we can see oxygenic similar to how plants do it, but there's also another totally different option where they don't use water as an electron source. So here are some images of some photosynthetic organisms. And, when you look at a chloroplast has an internal membrane. Here you're seeing internal membranes as well, even though these are microbes, right? So the purple bacterium on the right, these are small membranes that stack on itself. And then those, all these little kind of light gray dots, those are all membrane vesicles. And so those are usually the areas that you see all these pigments being embedded for photosynthesis. So even though we say, oh, prokaryotic organisms, microbes don't have internal organelles. Well, they certainly have a lot of like an organelle, but we tend to skip over that a little bit. So these microbes, they're prokaryotic, although there's all that stuff inside of them, we don't really say that those are organelles. Maybe someday they'll rethink that, but anyways. So that's where all the pigments are located, and then the complexes for doing the photosynthetic process. All right, so we said plants have two photosystems. Photosystem 2, photosystem 1. Microbes, so you have to back it up. Millions upon millions of years ago, before plants were even a thing, it was microbes doing photosynthesis. And we're going to see that some microbes, they only evolved one photosystem. That's what they put together. And it worked for them. So there are some microbes that just have one photosystem. And so they have photosystem. If they have photosystem 1, we tend to find that in green sulfur bacteria is often their nickname. And so what they're doing is they get an electron from some kind of external source like hydrogen sulfide or maybe an organic molecule laying around. It just depends. What's their environment? What have they evolved to have around them? And then they evolved that mechanism to strip electrons off of that. And it's the same idea where you'll have some kind of electron transport process, and then you'll have NAD plus usually sitting at the very end of the electron transport. It can be NAD or NADPH or NAD plus, I should say. It just depends on the microbe. And then that's good because you want the NADH or the NADPH so that they can make their sugars. So that's a really good thing. If they just have that one photosystem, they can still manage a way to get a proton gradient. It'll happen. And so as the electron transport chain process is going, you're going to get a proton gradient, which they use to make ATP. So these bacteria, they don't have a photosystem too. They have to just work with what they got with photosystem 1 and then a couple of other players in the electron transport chain process. So you tend to find these microbes, in general, they hang out in pretty dim conditions. They hang out in environments that are low light. And so they have to have kind of a ramped up ability to absorb light energy. So they have this chlorosome structure, we call it. And it's just like ramped up membranous material. So it's membranes, essentially. And it's the key thing that we're pointing to, these kind of white blobs that are pressed up against the cell membrane all the way around. So lots of chlorosomes, and they press up against the cell membrane because that's where all the action is in terms of photosynthesis. And so they, it's kind of interesting that even in really dim light, you can get pretty efficient photosynthesis going on, at least in the microbe world. And so dim light, you might think, why would a photosynthetic organism bother? But with this adaptation of the chlorosome, it really makes a huge, huge difference. So that's what they're trying to show here. That right picture is trying to show the chlorosome like hugging up to cell membrane. And then we have photosynthesis 1. And so you can see that there is an electron transport chain process here. It's just not super long, right? It's pretty truncated. You can see they use hydrogen sulfide, stripping electrons off of that. And if you do that, one of the big benefits is you get protons. But the other big benefit is, you're stripping electrons off and getting it into that electron transport chain. And then you see N plus sitting waiting to get that electron. And then it's the NH. So it's still an electron transport. You still have proton gradient. They didn't put the ATP synthase on. They omitted that part, but it's still happening. You get a proton gradient and then you get ATP synthesis. But this is pretty simple compared to what we saw initially with two photosystems. Pretty straightforward. And then that chlorosome structure is really, really helping. They're trying to show with that big squiggly yellow line that there's lots and lots of light energy getting absorbed. And then this is the same thing as what we just saw. It's just that they put it onto an energy reduction potential scale. And again, they're just trying to point out that once you absorb light energy, do that, you get excitation of electrons up into the negative scale, which just means it's going to be really easy now to donate that through the electron transport. And you can see paradox is involved. So that one's showing up again with the microbes that only use the photosystem 1. Let's compare that. So then there's other microbes that came along in the evolutionary process, and they're like, well, I'll evolve photosystem 2. but then that's where they stopped. It's like, okay, I got photosystem 2. I'll do my version of photosynthesis off of that. So that's all they have in terms of photosystems. And so we find those in purple. Purple non-sulfur bacteria is often their nickname. And so now this is odd because they use their pigment, their bacterial chlorophyll pigment, to extract electrons off of that. So what we mean is there's no ultimate electron donor external to the microbe. Like so far we've been saying, water, or hydrogen sulfide, or iron. This is actually part of the pigment of the microbe. So that's different. And so what we're going to see is that one of the electrons from that pigment will get excited because of light energy and get up into the negative scale of the reduction potential, and then it will do an electron transport chain process. So an electron gets taken off of the pigment, sent into an electron transport chain process. But another big difference is that then that electron circles back to the bacteria core. So when you follow the path of electron flow with these bacteria, it's circular instead of linear. It's a circular flow. That's another huge comparison to think about. Right? Circular electron flow. Everything else we've talked about to date, it's all been linear, where you have some, initial source of electrons, a final different source of electrons, and a linear path to get you there from A to B. This is an electron from bacterial chlorophyll, gets excited, goes around, comes back, gets excited again, goes around, comes back, gets excited again. It just zooms around and around and around. So it's kind of a cool little process. So they call this one cyclic photophosphorylation because the electrons are in a circular path. So cyclic photophosphorylation. One downside to this is that you can't directly make your reducing agent in this circular path. Kind of a bummer. So they don't make NADH or NADH directly. They have to figure out a different way to do that. Reductive biosynthesis just means that's getting your CO2 made into sugars. So let's just look at that as a comparison. So with photosystem 2, you're looking at this idea. You can see light energy striking photosystem 2. And they're trying to show that P870, that's the bacterial chlorophyll pigment. It's in there. It's in that region. And they're trying to show how the electron's coming off of that pigment. And then it's getting sent, if you follow the blue line, it's just going in a circle. And so you do have these donors and acceptors. It's pretty short. But it just keeps going round and round and round. By the time it comes back here, another one has been excited. This one fills the hole, so to speak, like from the hits it again, and it just keeps going round and round. And if you look what's going on here, you do see the artist did draw the ATP synthase. And yes, there is a proton gradient. Even though the flow of electrons is circular, you still get a proton. So that's good. But they don't show any plus anywhere if you're getting active. So the reason for that is trying to be alluded to here with this energy. So here they're trying to show that there's just not enough reduction potential to get NAD plus to become reduced. And so there's a process that the microbe has to kick into called reverse electron flow, which we've run out of time. I won't talk about it today. But We'll look at that next week. Okay. So make sure to ask questions if you have any via email. I'll be checking email between now and Friday. Let me know. And then I'll be here around 1:30-ish.